Okay, this is our last lecture. This is chapter 29, Development and Heredity. So we start with an overview of the prenatal period. That begins with fertilization. So the secondary oocyte and the sperm will unite, usually in the fallopian tubes. It ends roughly 38 weeks later with birth. It's composed of three shorter periods. We can break it down into the pre-embryonic, embryonic, and fetal. This should sound familiar from the lab pages. The pre-embryonic period is the first 2 weeks after fertilization. It is the zygote, which is the cell produced by fertilization. So once the sperm and egg come together, they form a zygote. It becomes spherical multicellular structure called a blastocyst. It just goes through a series of divisions. And it ends when that blastocyst implants in the uterine lining. Then you have the embryonic period, 3rd through 8 weeks. Rudimentary versions of the major organs start to appear, and then it is, at this point, it is called an embryo. Last is the fetal period. It's those last 30 to 34-ish weeks. The organism is now called a fetus, continues to grow and increase in complexity from what it started at, these rudimentary versions from the embryonic period. In the developmental period of pre-embryonic and embryonic is collectively called embryogenesis. So you start here with fertilization, where you form your zygote, and then it goes through just a series of divisions. And first, these are just fairly simple, just two cell stages. You get to the morula, which is the 16th stage, and then eventually you have your blastocyst, where you start to have differentiations of structure. Then we go into gastrulation, and this is, again, even just kind of more differentiation. You start to have some specific folding happening, some tubes forming. I'll go ahead and tell you, I'm not going to get super in-depth into gastrulation. It's pretty complex as far as types of structures and the way that development occurs. So we're going to look at it kind of basically. And then you have your organogenesis, where you have a lot of that development and increasing in size and complexity of the organs. So looking at fertilization, you have two gametes fusing to form a new diploid cell. Remember that these are both haploid. Your sperm and your egg are haploid, meaning that they have 23 chromosomes, and they come together to form a diploid with 23 pairs of chromosomes. This contains the genetic material derived from both parents and restores diploidy. It determines the sex of the organism, initiates cleavage, which is the divisions, and occurs in the widest part of the uterine tube. The oocyte is viable for 24 hours following ovulation, while sperm remain viable for 3 to 4 days. So this is why it is possible to, if you're trying to track ovulation specifically, your sperm are going to last a lot longer than the eggs will. Capacitation. This is a physiologic conditioning undergone by sperm to become capable of fertilizing the secondary oocyte. This will occur in the female reproductive tract. So you had spermiogenesis in the male system, and then we go through capacitation in the female system. Glycoprotein coat, and some of the proteins are removed from the sperm plasma membrane covering that acrosome. This lasts for several hours and allows that acrosome to be able to penetrate and digest the corona radiata on the oocyte. So millions of sperm are deposited in the vagina during intercourse, few hundred with a chance at fertilization. So that you have to have that many, because even though it can last for 3 to 4 days, not all of them will do that. They are attracted to the oocyte by chemicals that it releases. Progesterone released by cumulus cells around the oocyte bound to calcium channels in the flagella of the sperm, causing an influx of calcium, which is necessary for capacitation, the acrosome reaction, and fertilization. They attempt fertilization when reaching the secondary oocyte. Only one sperm is able to fertilize. The others will be prevented from entry. We'll look at that mechanism in just a moment. So the corona radiata penetration is the first phase of fertilization. There are 3. The sperm reaches the secondary oocyte, initially prevented entry by the corona radiata and zona pellucida, so you kind of have this wall that starts there. It can push through the cell layers of the corona radiata. And then when you get to the zona pellucida, you have this acrosome reaction. You're going to release digestive enzymes from the acrosomes on the sperm. It allows them to penetrate. They'll start to digest that zona pellucida and allow for penetration of the secondary oocyte. Once that happens, once you have, so multiple sperm can penetrate the corona radiata, but only one sperm will be able to penetrate the zona pellucida. So as soon as that happens, the zona pellucida will immediately harden and prevent other sperm from entering this layer. It ensures only one sperm fertilizes the oocyte. Um, and if two do get in, it's called polyspermy. If more than one sperm enters at the same time, it does not cause twins. It's immediately fatal because the organism will have 23 triplets of chromosomes, so it won't even be able to replicate. Okay, so once you have the fusion of the penetration of the membranes, you then have fusion of the sperm and oocyte membranes and pronuclei. So contact of sperm and oocyte plasma membranes, that'll cause them to immediately fuse, and so that only the sperm enters the oocyte. The secondary oocyte will then complete its second meiotic division. So remember, up until this point, it's been frozen in prophase 1, and then now it's going to be able to continue and finish up meiotic division and form an ovum. So only after the sperm enters nucleus, this kind of secondary oocyte finish its divisions and become an ovum, where it can then fuse their pronuclei. Each of those are going to have haploid number of chromosomes. Remember, they're only going to have 23 chromosomes. They fuse to form a diploid. And then you have a zygote. That is the single diploid cell that is formed. So once the sperm and the egg cell, once they go through the radiata penetration, the fusion of the membranes, and the nuclei, then you have what's called a zygote. That is the end of fertilization. So corona radiata penetration, acrosome reaction, penetrating the zona pellucida, and then fusion of the plasma membranes, which happens immediately, so that only the sperm pronuclei and the ovum come together. Okay, once that has formed, you then go through a series of mitotic divisions called cleavage. So the zygote is going to increase cell number, but not overall size. So it's going to stay within this kind of structure, here, once these two come in, it's going to stay inside that structure, but it will be increasing the number of cells within that. The size only increases after implantation in the uterine wall, which makes sense. You don't want to be increasing size in the fallopian tube. It's only so large. Before the 8-cell stage, the cells are not tightly bound together. They become tightly compacted after the third cleavage divisions. Compaction is the process by which contact between the cells is increased to the max, so they're literally just being pushed together. What does hold these cells together, so not tightly bound, but they are kind of interwoven together. What holds these cells together as they divide is fascia. So if you remember from 168, fascia is connective tissue that holds, generally, it's a generic term for connective tissue, right, really. And so you have at the cellular level, from the zygote, from the 1-cell stage to the 2, 4, 8, 16 cell stages, you have fascia that's holding that together. So when you see therapies like acupuncture, reflexology, trigger point therapy, all of those therapies are working off this idea that fascia connects our entire body. And so that's why you can work on the foot or work on the ear and affect the liver, the gallbladder, even physical skeletal and muscular structures as well. From there, you form the morula, which is the 16 cells. They continue to divide and develop the blastocyst cavity. At this stage, the preembryo is a blastocyst. It is formed from 2 layers, the trophoblast and the embryoblast. This will form the chorion. The embryoblast will form the embryo proper. These cells are pluripotent, meaning they're able to develop into any tissue. So the cells of the embryoblast are pluripotent, which they need to be. There is just a nice breakdown. There is the development as it travels to implantation. Okay, steps to implantation. The blastocyst enters the lumen of the uterus by the end of the first week, so usually about day 7. The zona pellucida around the blastocyst breaks down, and then it buries into the endometrium. This is when you have implantation. Beginning on day 7, the trophoblast is then going to divide into 2 layers, the cytotrophoblast, which is your inner, and then there's syncytiotrophoblast, which is your outer layer. After about 2 days, day 9, the blastocyst is completely buried into the uterine wall, where it contacts nutrients in the uterine glands for nourishment. The layer of the syncytiotrophoblast, from the trophoblast layer, it starts to secrete human chorionic gonadotropin, HCG. Signals the reproductive system that implantation has occurred, provides maintenance of the corpus luteum, which will continue to produce estrogen and progesterone to build the uterine lining. So it's a nice kind of positive feedback system there. Detected in urine by the end of the 2nd week. So most pregnancy tests are going to be testing for HCG. The levels are high for the first 3 months of the pregnancy, while the corpus luteum is still providing or while the placenta rather is developing and the corpus luteum is providing hormonal regulation. After that, though, it starts to decline because the placenta has started producing its own estrogen to maintain the pregnancy. There is a nice breakdown. You should be able to understand kind of the natural flow of hormones. You should understand, if you're given like a chart that says, oh, okay, HCG levels are here, progesterone and estrogen levels are lower down here. What stage of the pregnancy would you expect this to be in, early or late? You would obviously say, oh, HCG is only produced in the first 3 months. This must be early in the pregnancy. Or vice versa. If it was late in the pregnancy and there was still high levels of HCG, you would say something is not right. You should recognize that. Okay, chromosomal abnormalities and spontaneous abortion occur regularly during gametogenesis, fertilization, or cleavage. If they are severe, they result in spontaneous abortion, meaning if these chromosomal abnormalities are severe enough, they can result in a miscarriage. Many within 2 to 3 weeks after fertilization, before the pregnancy is even known. Perhaps 50% of pregnancies are terminated from spontaneous abortions. Half of those result from chromosomal abnormalities. Again, I know this is always a sensitive topic, and especially as a man, I can't really speak too much about this. But from my experience with students, it is something that in every class I have at least a couple of students who either have experienced this or have had other close friends or relatives experience it. And I think it's always important to talk about these things because there is definitely stigma associated to motherhood and being able to have children. And so it's always important as healthcare workers to understand how that can affect people emotionally, not to just, we have to understand that we treat people as a whole. They're not just something that they're going through. We have to understand how that affects them, might affect them socially as well. So this is something that is common enough for us to definitely be aware of. Okay, then you start to have changes to your embryoblast layers. By day 8, the cells of the embryoblast starting to form 2 distinct layers, your hypoblast and your epiblast. Lots of layers to remember in here, sorry. The hypoblast are layers adjacent to the blastocyst cavity, continuous with the yolk sac. The epiblast are continuous with the amnion, close to the amniotic cavity. They together form a flat bilaminar germinal disc. The bilaminar germinal disc and the trophoblast help to form your extra-embryonic membranes. They help to protect the embryo, mediating between them and the environment. They assist in nutrition, gas exchange, and removal of waste. The yolk sac is the first extra-embryonic membrane to develop, continuous with the hypoblast layer. It doesn't store yolk, as it does for birds and reptiles, but it's important for blood cell and blood vessel formation, early blood cell and blood vessel formation. So you should, just to kind of help you organize, because there's lots of things that we're going to be talking about here, lots of structures and things, you should definitely know what the extra-embryonic membranes are, what they do. The yolk sac is the first one to form. It comes from the hypoblast layer from the embryoblast. And it's important for early blood cell and blood vessel formation. If all you remember is that it doesn't store yolk, that's good. You should remember that, but that's probably not a question you're going to see. The amnion is continuous with the epiblast. It eventually encloses the entire embryo and forms the amniotic cavity to prevent desiccation. Specialized to secrete amniotic fluid bathing the embryo. Your chorion is your outermost extraonic membrane from the cytotrophoblast and the syncytiotrophoblasts. They blend with the functional layer of the endometrium, eventually forming the placenta, which is the site of nutrient exchange between the embryo and the mother. So for amnion, that's the only one. For amnion, it's from the epiblast. It forms the amniotic cavity, protects from desiccation, secreting fluid. The chorion is the outermost from the cytotrophoblast and syncytiotrophoblaст. It's blending with the functional layer to form the placenta for gas and nutrient exchange. Between embryo and mother, not from the outside world, but from the embryo and the mother. Your placenta is a highly vascular structure. It's the site of exchange of nutrients, waste, and respiratory gases between maternal and fetal blood, transmits maternal antibodies to the developing embryo or fetus, producing estrogen and progesterone to maintain the uterine lining. It starts during the 2nd week. So, basic properties. The fetal portion develops from the chorion. The maternal portion develops from the functional layer of the uterus. The connecting stalk connects the early embryo to the placenta, eventually forms the umbilical cord, which contains umbilical arteries and veins. Your chorionic villi, you should remember villi are finger-like structures formed from the chorion, containing branches of umbilical vessels. This is for gas and nutrient exchange between the functional layer of the endometrium with maternal blood vessels. The maternal blood does not mix with fetal blood. The blood streams are so close that nutrients and gases can mix, but you don't actually have a mixing of blood. O2 diffusing from maternal to fetal blood, while CO2 diffuses from the fetal to maternal blood. I'll let you tell me what is the driving factor for nutrient and gas exchange here. Placental characteristics, most growth occurs during the fetal period. It adheres firmly to the wall of the uterus. Remember, this is only going to be, it starts forming in the first 2 weeks, or by the second week it starts forming, but it does not finish until the 3rd month. So it's not until the 2nd trimester that the placenta has really grown. So most of that is going to occur during the fetal period, adhering firmly to the wall of the uterus, expelled from the uterus after the baby is born, called afterbirth. It's a selectively permeable structure for respiratory gases, allowing them to pass freely. Microorganisms and certain maternal hormones are prevented though, because it is selective. This, this is essentially basically just mammalian anatomy and physiology class, and so a lot of my experience is from veterinary medicine, and so they are watching animals give birth, and it is true for females as well, of humans. That expulsion from the uterus, it is you really have to kind of push it out. It doesn't. Sometimes I guess it comes out easy, but a lot of times it is just like another. Okay, now we have to push this out and get rid of the a lot of the afterbirth. And with animals, they eat it right there immediately, and part of that is because it's very, very nutrient rich. It has, for a dog, the one that comes to mind that certain ones stand out over the years was a little Dachshund. It was maybe 8 pounds, had 4 puppies, and so it was exhausted. And so as soon as it passed the afterbirth, it immediately ate it, and it gave it so much more energy that it was then able to go and feed its new puppies. We tend to stigmatize humans that want to retain their placenta and consume it in certain ways or use it for certain things. But it's important to remember there are, again, a lot of cultural things that go into maintaining the placenta. And birth is one of those. Birth and death are just those two such primal human experiences that it's really important to make sure that we're just respectful of how people handle those. Some harmful substances are able to cross the placenta, certain viruses, bacteria, drugs, alcohol, toxins in particular, that can cause birth defects or death. What's important to note is that dose and timing affect the fetus susceptibility. Pregnant women are urged to quit smoking, refrain from smoking, taking drugs, and drinking alcohol, definitely. We'll talk about this just in a little bit. So there's other formations of the extraembryonic membrane, you see you have this outer functional layer of the uterus right here that's going to eventually join with the chorion to become the placenta. You have your yolk sac, which thankfully it does not contain yolk. We remember that, but it is yellow. It appears yellow in this one at least. This is the first one to form. You have your amnion inside of your amniotic cavity, which actually contains the embryo. And then your placenta, which is going to form from these two. This is by early week 4. You can see that the yolk sac is starting to shrink, the amnion is starting to grow, as well as the functional layer of the uterus and the chorion. This is into late week 4. You can see, so now we're definitely in that embryonic period. We're starting to have some basic rudimentary structures being formed. You can see increased vascularization. And then we can get to gastrulation. So this is the critical period of development that occurs during the 3rd week. So this is late 4th week. So this is, again, you start to see this embryonic structures. Even from early week 4, there's a lot that's happening. So this makes it look like a lot of growth, and there is, we'll see. So this occurs during the 3rd week. And I only point that out because it's important to kind of remember where everything else is along in the process too. You still have your Syncytial trophoblast cells producing HCG to maintain the corpus luteum. The placenta is still developing at this point. In your embryonic period, you just started that. The epiblast is going to form 3 primary germ layers. Remember, a germ layer, a germ cell, is just a cell or a layer from which other things develop. This is going to be your ecto, meso, and endoderm. The 3-layered structure eventually is called an embryo. So once you have these 3 layers, these 3 separate layers, that's when you're in your embryo. Gastrulation begins with the formation of the primitive streak. It's a thin depression on the surface of the epiblast. You have your primitive node, which is the cephalic end of the streak, up towards the head, consists of the elevated area surrounding the primitive pit. During invagination, the cells detached from the epiblast layer migrate through the primitive streak between the epiblast and hypoblast layers, so it's kind of going in between. The epiblast is the source of those 3 primary germ layers. All body tissues and organs derive from these. The endoderm displace the hypoblast cells. The mesoderm are a new primary germ layer formed by the epiblast cells. And the ectoderm are the cells remaining in the epiblast. Okay, so this is early week 3. You can see you have your caudal, which is your tail end, your cephalic end up here along the primitive streak. In the middle, you have cells that are going to be going in and between these layers. So there is your yolk sac. This is going to be, let's see, how do they label this? They label it so they label this as the amnion. And then right into here, you have the cells of the embryo that are going to be forming. So they go in and then divide between, and you start to have your hypoblast, your epiblast layer right up here, and then this invagination of these epiblast cells in between. That eventually formed your endoderm, your mesoderm, and your ectoderm. Okay, so then we get into folding of the embryonic disc. The embryonic disc is a flattened disc-shaped 3-week embryo, so basically what we were just looking at. It starts to fold on itself during the late 3rd and 4th weeks by cephalocaudal and transverse folding. So cephalocaudal is head to tail, transverse is side to side. And it's exactly what it sounds like. It's just going to be folding and folding and folding and folding. Occurs, so cephalocaudal folding occurs in cephalic and caudal regions of the embryo. Rapid growth of embryonic disc and amnion, no growth of the yolk sac, causes head and tail to fold on themselves. So it starts to go from this kind of flattened disc into making layers of itself. Transverse, or the left and right sides of the embryo, converge towards the midline, start to pinch off the yolk sac, fusing sides of the embryonic disc in the midline, creates a cylindrical embryo. Ectoderm solely along the exterior of the embryo. Endoderm confined to internal region, and it creates the torso region. All right, so what I want you to be able to know, based for the 2 types of folding, you should definitely know kind of what direction it's folding in. You should know that for the transverse, it starts to separate the ecto and endoderms and creating your torso regions. And that's good. Differentiation of the ectoderm on the external surfaces of the cylindrical embryo, responsible for forming the nervous system tissue, called neurulation. Forms epidermis, sense organs, pituitary gland, adrenal medulla, enamel of the teeth, and lens of the eye. So you should know the layer, the ectoderm, where it's located, external surface of the cylindrical embryo, what it's responsible for, the basic systems that it forms, and the basic structures that it forms. So this is your important side. The folding, really this is a concept that I want you to understand. I want you to understand that you go from this kind of flattened disc here, and then after a series of cephalocaudal folding and transverse folding, you start to make a cylindrical structure that's going to continue to develop in complexity. And it's going to start to have differentiation of these 3 layers. And so this is the really kind of important part. The concept of folding you should know to kind of create a 3D embryo, but then the differentiation of the layers are what we're going to get into. The mesoderm There are 5 categories. You have the notochord, formed by tightly packed midline cells. It's the basis for the central body axis and axial skeleton, and induces formation of the neural tube, which eventually does become your CNS. The paraxial mesoderm forms on either sides of the tube, forms somites that form the axial skeleton, muscle, cartilage, dermis, and connective tissues. Keep on going. The intermediate mesoderm forms most of the kidneys, ureters, reproductive system. The lateral plate forms the spleen, adrenal cortex, cardiovascular system, serous membranes, and connective tissues of limbs. And the head mesenchyme forms connective tissue and musculature of face. I'm not going to ask you, know the names. Whether or not I ask you them specifically is yet to be seen. I probably won't ask you for these specific names, but you should know the different structures that the mesoderm is forming. That's just differentiation of it. And then the endoderm. So this becomes the innermost tissue, so innermost after the transverse folding, forms the linings of the GI, respiratory, urinary, and reproductive tracts, the tympanic cavity and auditory tube, the liver, gallbladder, pancreas, palatine, tonsils, thyroid, parathyroid glands, and the thymus. So the 3 periderm layers and their derivatives, I can't remember what this YouTube link is for. I'm guessing it's probably something about differentiation of the germ layers, so I would definitely watch that when you have the chance. But this is kind of really going to be a helpful slide for you because it gives you the layer and the structures that it's associated with and that it forms, and that's going to be the most important thing for me. Okay, so organogenesis. This begins around week 8. Sorry, 1 second. This begins by week 8. You have upper and lower limbs that have adult shape, and most organ systems have their rudimentary form, particularly sensitive to teratogens during this time. Substances causing, I think I said that wrong. This ends by week 8. You have organ development. Particularly sensitive to teratogens, which are going to be your alcohol, tobacco, toxins, things like that, that cause birth defects or death. So peak development are at different times for different structures. So your limbs, their peak development are weeks 4 to 8. The external genitalia, the late embryonic, occurs through the late embryonic through early fetal period. So teratogens are most dangerous during the peak development of a particular system. So let's say you weren't aware that you were pregnant, were consuming drugs and alcohol heavily during the weeks 4 to 8. This could impair their limb development, their external genitalia development, as well as any other structures that happen to be occurring during that peak time. There are some structures that only, their development is limited to a certain week, maybe week 6, and it could be that if you were abusing during that 1 week, or you got, I keep saying abusing, but it could be a virus, it could be you get exposed to something in those early weeks when that particular system is developing, it's going to be most hardest affected. Then you have the fetal period from the beginning of the 3rd month to birth, maturation of the tissues and organs, rapid growth of the body. The length of the fetus is measured as crown to rump length, or crown to heel. 2.5 centimeter embryo grows to an average of 53 centimeters at birth. Weight increase is most striking during those last 2 months of the fetal period. Infertility and infertility treatments. This is the inability to conceive and maintain a pregnancy. Either inability to conceive can be male or female systems. Multiple causes, including blocked uterine tubes, bipelvic inflammatory disease or endometriosis, ovulatory disorders, anti-sperm antibodies or low sperm count, abnormal sperm, they have low motility, impaired sperm delivery. I think that's supposed to be erectile dysfunction. Or, if you just do not have a strong ejaculate, if you aren't producing enough prostaglandins or alkaline bicarbonate to help reduce the acidity in the vagina, then the sperm aren't going to be able to survive. Multiple possible treatments, intrauterine insemination, oral medications, in vitro fertilization, donor oocytes. Luckily, we live in a world where there are a lot of ways to become a parent. Okay, pregnancy is divided, and now we can look at a little bit bigger source of pregnancy. So divided into 3 trimesters. The first is the first 3 months of the pregnancy. Shocking. The zygote, starting with that sperm and egg cell, becomes an embryo and then an early fetus. By the second trimester, this is when the placenta will be fully developed, remember? So these are the months 4 to 6 of pregnancy. Growth of the fetus and expansion of maternal tissues. And then the third trimester is the months 7 to 9. You have rapid growth of the fetus, the mother's body preparing for labor and delivery. Okay, hormonal changes during pregnancy. Estrogen and progesterone to start with. Those are the big regulatory ones here. They're produced by the corpus luteum during the first trimester, and the placenta in the second and third. High levels will suppress FSH and LH secretion to pause ovarian cycle and follicular development. Doesn't always occur. Sometimes you can still ovulate while you're pregnant. These hormones help to facilitate uterine enlargement, mammary gland enlargement, and fetal growth. It also allows for faster growing nails, fuller hair, relaxation of ligamentous joints, and it stimulates the uterus functional layer to grow. Progesterone does that. Relaxin. This is secreted by the corpus luteum and the placenta to promote blood vessel growth in the uterus. It is called relaxin because they used to think that it aided in the relaxation of ligamentous joints, but it doesn't. That is estrogen and progesterone. Relaxin is to help promote blood vessel growth in the uterus. So if you're confused about why it's named that, there you go. Corticotropin releasing hormone is secreted from the placenta in large amounts, role in the length of pregnancy and the timing of childbirth, responsible for aldosterone rise in the mother to promote fluid retention and edema. Human chorionic thyrotropin is from the placenta as well, stimulates the thyroid gland to increase the mother's metabolic rate. Human placental lactogen from the placenta once more affects how the pregnant woman metabolizes certain nutrients. The mother metabolizes more fatty acids instead of glucose, inhibiting the effect of insulin to allow more glucose to be available for fetus. I think we have a slide coming up here in a second about gestational diabetes. Prolactin, increased levels 10 times by the anterior pituitary to ensure lactation occurs after giving birth. Oxytocin delivered increased levels produced by the hypothalamus. Remember that oxytocin is produced by the hypothalamus. It is stored in the posterior pituitary along with ADH. Oxytocin is involved in uterine contractions, milk letdown from the mammary glands, and it really starts to increase in the second and third trimesters in response to those rising estrogen levels, again in preparing for childbirth. Okay, uterine and mammary gland changes. The uterus will expand and begin to enlarge once implantation occurs. By 12 weeks, the uterus is just superior to the pubic symphysis. It impinges on the space of the bladder, causing more frequent urination, particularly during the first and third trimesters. Most enlargement is due to muscle hypertrophy, hyperplasia, placental growth, and amniotic fluid. This video is a kind of quick, like rotating. I don't think it's a GIF. I think it actually is just a video, but it shows the development of the fetus and the changes of kind of represents the changes that internal organs go through pregnancy. It's pretty interesting. The mammary glands, they become tender during the first trimester due to the increasing estrogen and progesterone levels. You have melanocyte stimulating hormone from the placenta, which causes a darkening of the areola and the nipples. It darkens the linea alba, which runs down the center of the abdomen and becomes the linea nigra. Growth of mammary glandular tissue, development of additional acini in the mammary glands. So there you can kind of see how it starts off very low, very small, and then rapidly starts to expand to impinge on all the organs. No wonder it's uncomfortable. Digestive system, nutrient, and metabolic changes through pregnancy. So we already said that increased insulin resistance occurs during pregnancy due to increased levels of corticosteroids, estrogen, progesterone, and human placental lactogen. It can lead to gestational diabetes in the mother because you just have these decreased sensitivity to insulin. Morning sickness experienced by many pregnant women during the first trimester particularly. Doesn't just occur in the morning, some with a little, some with really severe symptoms. Causes unknown, possibly due to high hormones. From what I've heard, generally it gets better after the first trimester, but that is certainly not the rule or the case for all. Okay, you get abdominal complaints during pregnancy. Slowed intestinal motility, constipation is very common, can lead to hemorrhoids if they're struggling to defecate. High progesterone results in relaxed, smooth muscles. Materials remain in the GI tract for longer periods of time. You have abdominal organ compression occurring, resulting in heartburn and indigestion. Weight gain occurs because the fetus is growing. The placenta, breast, and uterus are developing and creating more cells. You start to retain more fluid. We saw that aldosterone levels increase. And adipose tissue increases. Once the body starts to get into this kind of state, it's going to start trying to store and hold on to as much as it can to provide extra nutrients. So the body knows that it needs to have this extra sources on hand, energy sources on hand. You take in about extra 300 calories a day is needed. Require adequate folic acid, calcium, protein, and iron. So most pregnancy vitamins. Okay, gestational diabetes develops during pregnancy. Increased insulin resistance and high blood glucose usually appears in the second trimester. May develop high blood pressure and complications. You increase the risk of a larger baby, increased risk of needing a C-section and birth complications, as well as hypoglycemia. Special diet to regulate your blood glucose, and then that does put you at a higher risk for type 2 diabetes later in life. Hyperemesis gravidarum, severe nausea and vomiting during pregnancy, causes dehydration, electrolyte imbalance, and weight loss, may require hospitalization and IV fluids, relatively rare, thankfully. Hyperemesis, if you remember, emesis is vomiting. Okay, cardiovascular and respiratory system changes then. So the cardiovascular system undergoes dramatic changes during pregnancy because of the extra blood that's needed and the distribution of respiratory gases to the mother and the fetus. The plasma volume will increase by 50%. Hematocrit then becomes lower because you're increasing your plasma content, your water content. The cardiac output will increase by 30 to 50% to make up for that. And the heart rate and stroke volume will increase as a function of cardiac output. Blood pressure may initially increase during the first trimester and then later drops due to decreased peripheral resistance. Compression of the abdominal blood vessels by the fetus can impair venous return from the lower body, causing varicose veins, hemorrhoids, and edema. Preeclampsia is high blood pressure occurring by the second half of pregnancy. Diabetes, older age, obesity, previous to preeclampsia. Cause is generally unknown. General risks of hypertension for mother. The placenta will then have poor perfusion. Giving birth is the only real cure. Maybe prescribe bed rest medications or labor induction. Usually they'll just kind of prescribe very extreme bed rests until it is time, usually until at least the 38th week when there's, or however long they can push it before inducing labor. True eclampsia is high blood pressure causing seizures, which is a medical emergency, obviously. The respiratory system, what happens is that expanding uterus prevents the diaphragm from fully descending and the lungs from completely expanding, which can cause dyspnea, uncomfortable awareness of breathing, and may result in epistaxis due to increased circulation, that increased blood pressure. Progesterone increases the brain's sensitivity to carbon dioxide to help lower the blood CO2 levels. That's going to facilitate diffusion of gases across the placenta. So it will cause the mother's blood CO2 levels to drop. So instead of being, at our standard of in systemic cells, 45, it may be lower at 40 or 42. And then since the placenta cells will still be at their standard of, I believe, 45, that concentration gradient will be lower moving towards the mother. So that way it just ensures that CO2 diffuses out of the placental cells. Providing oxygen in the mother to the fetus, it will increase tidal volume, pulmonary ventilation, respiratory rate, and oxygen consumption. That increased calories comes from it comes from the need to increase metabolism. Okay, the urinary system eliminates waste from both the mother and the fetus. The fetal waste does not just sit in the amniotic fluid. That's not what it's for. It must filter 50% more plasma volume, so your GFR increases by 30 to 50%. You have dilation of the ureters and renal pelvis, can result in urine stasis, so it just kind of sits there. Ureters sometimes get compressed by the uterus, causing urinary tract infections to be more common, as well as more frequent urination. Okay, factors that lead to labor. We're flying through these slides. Okay, I always have to make sure it's recording. Okay, so labor is the physical expulsion of the fetus and placenta from the uterus, typically at 38 weeks to 42 weeks for full-term pregnancy. Not all uterine contractions lead to true labor. They begin weak and irregular at first, can be noticed as soon as the second trimester. You have those oxytocin levels starting to increase, but they become more and more frequent as estrogen and oxytocin levels increase. Premature labor is labor prior to 38 weeks. Undesirable because the infant's body system is not fully developed. From what I've heard and from what I've seen, viable is usually about 23 weeks is the earliest that most a fetus can be viable. My cousin works in the NICU, and so she sees a lot of babies at 24, 25 weeks. So earlier than that, any kind of premature labor or premature birth has its risks for sure. False labor, the uterine contractions not resulting in the full labor. Braxton Hicks contractions are regularly spaced and do not become more frequent. Relatively weak. Pain is limited to the lower abdomen and pelvic region, not the entire abdomen itself. Pain sometimes stops with movement and does not cause any cervical changes. So true labor is the uterine contractions that increase in intensity and regularity and result in changes to the cervix. The mother's hypothalamus secretes increasing levels of oxytocin. The fetus also secretes more oxytocin. And then both sources stimulate the placenta to secrete prostaglandins. If you remember, those are fatty acids that act like a local hormone. They stimulate uterine muscle contraction and dilate the cervix. Combined maternal and fetal oxytocin initiates your true labor. So oxytocin from the mother and fetus will stimulate the placenta to secrete prostaglandins that will stimulate muscle contractions, dilating the cervix, and the oxytocin will also see has roles. So true labor characteristics is an increase in frequency of contractions over time. They're not staying evenly spaced out. They're usually getting closer and closer, as well as increasing in intensity. Pain radiates from the upper abdomen to the lower back, not going away in response to movement, and the contractions facilitate the cervical dilation and eventually expulsion of the fetus and placenta. We'll see this can take anywhere from, 20 minutes to 20 hours. Okay, true labor is a positive feedback mechanism. The intense contractions pushes the fetus's head against the cervix. So the position of the fetus is also very important, and what will tend to happen is that as the fetus is preparing for birth in those last few months, it will start to flip and move its position so that way its head is up against the cervix, hopefully. Not always, but hopefully. So as, assuming that the fetus is in the proper position, as the uterus is contracting, it's pushing the fetus's head up against the cervix, which stimulates that to stretch and dilate. If you remember that property of smooth muscle where it will stretch, and then if that stretch is maintained, it will relax and not contract back on itself. It signals the hypothalamus to secrete more oxytocin, stimulating the placenta to secrete more prostaglandins. So the fetus pushing against the head of the cervix does all of these things. This causes more intense uterine contractions. All of these will. Let me say the oxytocin and the prostaglandins will cause more intense uterine contractions. This continues to intensify until the fetus is expelled. With expulsion, the major source of prostaglandins is removed. The uterus and the cervix are no longer being stretched. You have a drop in oxytocin levels, and labor will cease. So here is just a great little representation of it. You should understand stimuli, control centers, and effectors. Understanding that it is a positive feedback mechanism. Okay, so inducing labor is recommended if you are 2 weeks past your due date. Um, you will be hospitalized the night before. Not sure how true that still is, because it's become such a common thing. I'm not sure how true that still is. Prostaglandin gel is administered to help assist with cervical dilation. An IV synthetic oxytocin is given to initiate true labor. They perform several nerve blocks. The pudendal nerve block numbs the pudendal nerve, which is the main sensory nerve of the perineum, to numb the lower vagina and the perineum. You can feel contractions and the vaginal stretching. The epidural nerve block is placed in the epidural space of the spinal cord. It numbs the uterus, vagina, perineum, and lower limbs to some extent. Relieves the pain associated with contractions. Usually does not interfere with contractions themselves, though those will still occur. The spinal nerve block is reserved for cesarean sections. The anterior abdominal wall is numbed prior to any incisions, and the limbs and pelvis are completely numbed at this point. Okay, looking at the stages of true labor, 3 stages. Your dilation stage is the first. It begins with the onset of those irregular uterine contractions, ends when the cervix is effaced, which means thinned, and dilated to 10 centimeters. It is the longest of the 3 stages, meaning that it's going to have the greatest variability in how long it's going to take. Nulliparous women who haven't given birth usually have a longer dilation stage. Parous women who have given birth may clear it in 4 to 12 hours. Again, it can happen very quickly. The dilation stage starts with regularly spaced uterine contractions, increasing in intensity and frequency. The baby's head against the cervix causing effacing and dilation. You have this rupture of the amniotic sac, which is your water breaking, and can be manually ruptured if necessary. So if the dilation stage has been going on, but the water still hasn't broken, but you can clearly see that the cervix is starting to efface, it can be important to do that because the fetus might not be able to expel fully if it does not. So in this one, you can see how the cervix is partially dilated, that entrance to the vagina is partially dilated, whereas over here, it's 10 centimeters to allow some accommodation for the head of the fetus. The expulsion stage begins with the complete dilation of the cervix and ends with the expulsion of the fetus, usually 30 minutes to several hours. Nulliparous women definitely taking longer, not definitely, but typically. Uterine contractions help to push the fetus through the vagina, facilitated if a woman bears down using that Valsalva maneuver, which is where you close the epiglottis over the larynx, and the trachea, and you can bear down and increase your abdominal pressure. Crowning is when the first part of the baby's calvarium distends the vagina. The head is followed by the rest of the body. An episiotomy is occasionally necessary where the perineal muscles are surgically incised to create a wider opening. This is just to prevent tearing, because tearing is typically a jagged line, which is harder to suture together. So they go ahead and will cut a clean line to make it easier to stitch together. The umbilical cord is clamped and tied off. So there you have your crowning. At this point, you have expulsion. Okay, I'll stop at this real quick. True story, had an animal come into the vet's office that had, it was another Dachshund that had been bred accidentally with a larger dog. They don't know what it was, but it only had 2 puppies. We did an x-ray, it only had 2 puppies. But when they brought it in, the puppy's head had been expelled from the vagina of the dog, but its shoulders were so broad that it couldn't actually get back through. It couldn't expel the rest of the shoulders. So we kind of had this situation where we had a puppy that was partially expelled, that had been partially born, and was still alive. And was, this was pretty quickly, the owner saw that after half an hour or so, nothing was happening. And so they brought it in smartly. But it was very strange. One of our stranger days. If you want to know how that story ends, you can message me privately. The placental stage occurs after the baby is expelled. The uterus is continuing to contract, compressing the uterine blood vessels, which will help to displace the placenta from the uterine wall. Remember, it's ingrained and continuous with the functional layer of the endometrium. So you're having to shed all of that. After birth, the placenta and remaining fetal membranes. Expulsion is usually completed within 30 minutes. Carefully examined to make sure all is expelled because complications can occur if fragments are left. Yes, there is your placenta, your expulsion of the placenta. Remember, it's having to kind of come out from the endometrial layer. Okay, fetal positioning in the dilation stage. Normally the fetus is in the vertex position with its head down facing the sacrum, so facing backwards. This is the ideal position for dilating the cervix and pushing the fetus through the vagina. If the fetus has been breached, that means that its buttocks are coming first. That can delay the cervical dilation because you don't have that hard, kind of almost battering ram head. Variant positions for extraction, forceps, vacuum possibly if needed. They actually take the forceps, put it right around the fetus head and pull it out. You could do a cesarean section where they're actually delivered through an abdominal incision. Okay, postnatal changes for the newborn. Respiratory changes in the neonate. The fetus, after being expelled from the uterus, is called a neonate. The fetal lungs not fully inflated prior to birth. Takes the breath, but the fetus will take, or the baby, the neonate at this point, will take their breath within the first 10 seconds of birth, caused by the central nervous system reactions to changes in the environment. The lungs are inflated with that first breath. Surfactant is being produced to keep the alveoli open. If they're born earlier than 28 weeks, their surfactant is going to be insufficient, and they may need a ventilator to help maintain their lungs until they mature fully and can produce surfactant themselves. So prior to birth, the blood is shunted away from non-functional lungs. So the ductus arteriosus and the foramen ovale and the heart are going to just make sure that as blood comes in through the umbilical arteries, umbilical veins, that oxygenated blood in through the umbilical veins is going to just be going from the heart directly into systemic circulation in the fetus. But with the first breath, you have a drop in pulmonary resistance, a dilation of the pulmonary arteries, and the pressures are going to switch. So you have a drop of pressure on the right side and an increase in pressure on the left side. Postpartum is the time period after giving birth. So also this is the change in the mother now. The woman's body continues to undergo further changes to both feed the neonate and to return to pre-pregnancy form and function. Estrogen and progesterone levels start to plummet within a few days after birth. The uterine lining is no longer needed. This can also cause shedding of what's called that losha. So for days, weeks, months after birth, there can still be some production of what they call the losha, which is just parts of the uterine lining continuing to shed. See your doctor if it lasts for too long. Feelings of sadness and depression, called the baby blues. You have this plummeting of hormones. The hair reverts back to normal. Hair loss cycle. You have this peak in hair loss 3 to 4 months after delivery. Your chemoreceptors are less sensitive to the carbon dioxide. They go back to their higher levels. So your respiratory rate, tidal volume, and pulmonary ventilation return to normal. I always say it's just kind of you just kind of get hit. You go from having a baby, now you're probably tired trying to take care of that new infant. You're possibly having to deal with any complications from afterbirth, and then you start to already get sad and depressed just from a natural hormonal process, and then your hair starts to fall out. So it really just kind of, we always need to take a moment to praise mothers. Okay, corticotropin releasing hormone, CRH, levels will drop dramatically. The placenta stops producing CRH. High levels during pregnancy associated with postpartum depression. Severe depression needing immediate treatment. This is not something to joke about. This is definitely something that is very serious and should be talked about, should be addressed. Checking in on normal mothers, making sure they have support systems and people to talk to that can notice any types of changing behavior. Blood volume and fluid changes. So you start to have this fluid reduction. That additional fluid was being retained during pregnancy. It does require quick and efficient expulsion. The amniotic fluid is expelled during labor. The losha is a portion of blood volume, mucus, and endometrial tissue expelled via the vagina. It's heaviest in the first 5 days and continues for several weeks, as I was saying. Could be longer, hopefully not for you. You have excess fluid being expelled through urination, declining aldosterone levels. That precipitates an overall drop in your blood volume. And you have that increased urination in the first 24 hours. Profuse sweating is also common, trying to get rid of extra fluid. Lactation then. So lactation is a production and release of breast milk from the mammary glands. Your prolactin from the anterior, prolactin is produced in the anterior pituitary. It is responsible for your milk production. Its secretion is inhibited by dopamine in non-pregnant women and in men, increased by high levels of estrogen. So both estrogen and prolactin cause the acinar proliferation and branching of the lactiferous ducts that move through the mammary glands, responsible for preventing breast milk secretion until after birth. All right, so the first fluid produced is not milk, it is colostrum. During late pregnancy and the first few days after birth, it is a watery milk-like substance. It has a lower concentration of fat than true breast milk will have, but it's really rich in immunoglobulins, particularly IgA, allowing for quick passive immunity from the mother. It also causes a laxative effect, facilitating the infant's first bowel movement, which is probably needed and colorful, we'll just say. Breast milk starts to be produced for the first few days postpartum. It has that higher fat content than colostrum. It also has essential fatty acids, enzymes for digestion, as well as further immunoglobulins. It is more easily digestible than breast milk substitutes and optimal source of nutrition for an infant. Again, lots of opinions and personal, cultural things that go into breastfeeding. Some people have a harder time with it than others. Some people do not have the time to do it, depending on how they have to, if they have to go back to work quickly enough. So just being able to present any kind of clients, any kind of patients that you may have, with the idea that it is more easily digestible. It is a more optimal source. There was a large movement to move towards formula. I was a formula baby. Grew up just fine, but there's science. That's what we're here to talk about. So milk letdown is the release of breast milk. It involves positive feedback mechanisms, so meaning that all of these stimuli are going to increase secretion of milk letdown. Suckling mechanical receptors are stimulated in the breasts that send signals to the hypothalamus to produce oxytocin, which is then released from the pituitary, targets myoepithelial cells in the mammary acini. The cells will contract, releasing breast milk from them. As milk is released, the infant continues to nurse and suckle, which facilitates that whole response over again. So again, you should know the stimuli and what it's going to be doing, whether it's inhibiting or stimulating. Okay, so prolactin spikes occur in prolactin production each time the baby breastfeeds. It promotes new breast milk production, and as the infant feeds, dopamine release is inhibited by the hypothalamus. That stimulates large amounts of prolactin secretion to make sure that there's plenty of milk for the fetus, for the baby. Inhibition of ovulation often occurs with regular breastfeeding. GnRH release is inhibited from the hypothalamus. Without that, FSH and LH aren't released to help prevent ovulation. It is not a reliable form of birth control. I made this larger and more bold. They did not create this slide like this, but I just want to make sure we point that out, that it is not. Okay, after pains. Contractions of the uterus after giving birth stimulate shrinkage of uterus to close to its pre-pregnancy size, stimulated by oxytocin, less severe after the first week, facilitated by breastfeeding. Okay, so now we get into genetics. So that was pretty much all of our pregnancy development stuff, so now we can do more genetic things. So basic definitions that you should know: heredity, transmission of genetic characteristics. Karyotypes are the actual display of the chromosome pairs, like you would see from, like, if you were to do like a scan of them. Homologous chromosomes are paired chromosomes with genes for equivalent biological characteristics, meaning that they're going to code for the same type of thing, either tall or short or green hair, blue eyes, whatever. And then your autosomal cells, your autosomes are your 22 pairs of chromosomes without genes determining your sex, your 1 through 22. Here's your karyotype. You have this is your autosomal cells, you can see are all numbered, and then you have your X and Y cells over here, your X and Y chromosomes. Your sex chromosomes are the last two containing genes that specify your sex. A locus is a specific space where each gene is located on a chromosome. So, a locus, it could be for however many base pairs that gene is. It's like if it's your gene, if it's the gene that codes for, um, we're just talking about it, proper prolactin synthesis. That gene could be 15 base pairs long. It's going to be on the same place in all of that chromosome. Whenever you look at any person's chromosome, it's going to be on that same spot. The alleles are variants of one gene found at some locus on homologous chromosomes. Dominant allele will be expressed or physically shows the trait, represented generally by a capital letter. Recessive trait is masked, expressed only if present on both chromosomes, homologous chromosomes. Punnett squares are boxes showing gene combinations. Homozygous if you have the same alleles. Heterozygous if you have different alleles. Genotype is your genetic makeup. Phenotype is your physical expression. There's dominant versus recessive, so just depending on the particular traits. We did a lot in that lab. Okay, patterns of different inheritance. You have your strict dominant recessive inheritance, which is also known as Mendelian inheritance. The dominant allele always expressed in the phenotype. Relatively few traits actually follow this pattern. So Mendel was working with pea plants, talking about tall or short, or wrinkled leaves versus smooth, or wrinkled peas versus smooth peas, those kind of things. And those generally are, for a simple plant and simple things like that, coded for by very few alleles. But that's not always the case. Usually things that we think of as phenotypes or any kind of portions of our body are coded for by multiple genes. May be affected by environmental factors. Can't remember if we talk about epigenetics or not, but nurture does have an effect. So incomplete dominance, so this was strict dominance recessive. One, if it's dominant, it will show. If recessive, it will not show, unless it's homozygous recessive. For incomplete dominance, you have two heterozygous alleles. The phenotype is an intermediate between the dominant or recessive. So sickle cell is one of those. Most individuals with two identical dominant alleles coding for normal hemoglobin. The sickling allele produces abnormal hemoglobin. And so sickle cell disease is, you have full sickle cell disease if you have two homozygous recessive alleles. Heterozygous individuals carry the sickle cell trait, under low oxygen conditions. Some erythrocytes may develop the sickle shape. So essentially, if you are homozygous dominant for it, you aren't going to show any of the trait. If you are homozygous recessive, you are going to show the full sickle cell trait. And if you are heterozygous for it, then you will have some variation of it at certain circumstances. Codominance, your two alleles are equally dominant, both expressed in the phenotype, such as your ABO blood group. So both A and B are codominant. The third allele, I, is the recessive for your O blood type. Chapter 18. Polygenic inheritance, multiple genes interacting to produce the phenotypic trait. Most of our traits will result from this. Genes on same or different chromosomes, eye color, height, skin tone, predispositions to many diseases are polygenic inheritance. Your sex-linked traits are expressed by genes on your X or Y chromosomes. 900 to 1400 genes on the X chromosome, most not involved in any kind of sex determination. 70 to 200 on the Y chromosome, mostly for development of male characteristics. Sex-linked traits most often involve your X chromosome. There's just more of them. X-linked traits are always expressed in a male because they only have one X chromosome. Only expressed in the female only if she has two recessive alleles, which is a pretty low probability. A carrier is a woman with one X-linked recessive allele, doesn't exhibit the phenotypic effects, but can pass that X-linked allele to their children. If passed to a female, they also become a carrier. If passed to a male, they'll express that X-linked trait. Color blindness. The individual will have trouble distinguishing red and green. Women are rarely colorblind, requiring both recessive alleles. They are typically carriers. Men will inherit the allele, lacking the normal one to counteract that color blindness, and so they will exhibit the colorblind phenotype. Hemophilia A is a disorder of blood clotting. Individuals with disordered will bleed profusely after some type of injury. Again, females are carriers, males are affected because X-linked. There's just a nice Punnett square. You can see that there is a chance that you would have a normal son, but also 25% chance of a carrier daughter and 25% chance of a son with hemophilia. X-linked dominant traits are relatively rare, expressed in both males and females who carry it. Men are typically more severely affected. They have no normal recessive allele to counteract the effects. Many male zygotes with X-linked dominant disorders are spontaneously aborted. Okay, penetrance and environmental influence on heredity. Penetrance is the percentage of a population with a genotype exhibiting expected phenotype. This is influenced by a variety of factors. But essentially, it's just saying, okay, this is, if people have this trait, we expect them to have this type of, or if they have this gene, we expect them to have this phenotype or this trait, and then how much of that is actually present in the society of people with that trait or with that gene. Environment can have variable influence on many genetic traits. Teratogens can cause harm in inheritance and interfere with phenotypic development, such as fetal alcohol syndrome. Poor nutrition can also have a negative effect on development, and certain alleles will increase your risk of cancer development. So real quick, so epigenetics is essentially what this is referring to. You have your standard DNA, but there are markers, there are certain proteins that are activated or deactivated by age, by diet, by exposure to environmental factors such as sunlight or pollutants, things like that. There are certain environmental things in how we live our life and how we're raised that will activate or deactivate other genes. So that's why you can have certain alleles with risk of cancer development, even if you don't have a history of cancer in your family. If you, for some reason, have a completely different lifestyle than your family, or someone that has a very healthy lifestyle, you're like, wow, how'd they get cancer? Look how healthy they are. Maybe something in that lifestyle that they were living actually activated certain genes, or it was just a time thing. There's and one thing that I do always think is interesting is like the science of death. I know this whole thing was about birth, but looking at death and why we still die, why we haven't been able to evolve past that yet. And it's because certain traits, certain diseases aren't activated until later in life, beyond when we've already, at that point we have already typically would have, had children, passed down our genes. And so we're passing down these genes that have disorders, things like Alzheimer's and dementia, Huntington's, that you're able to live a normal life until you go through your reproductive years, and then it affects you, and then it starts to cause degeneration. We continue to pass on those traits and those genes, and again, some of them don't arise until there is the specific environmental factors for that.